Welcome to the SINGEL Recordings, a podcast series in which we share remarkable moments of reflection, conversation, and debate that have taken place on our stages. In this 7th episode, you will hear Anna Tsing. At the invitation of the SINGEL and the Flemish Institute of Architecture, she discusses the future of fungi and the insights they offer into the challenges of our time. This lecture and interview was recorded in the SINGEL's theater studio on the 26th of May 2026.
Good evening. My name is Tim Taverne, and I am the curator of the Talks and Debates program here at the SINGEL. First of all, I would like to thank you for being here. Tonight is the final talk of the season, and I couldn't imagine a better speaker to close the series this year. It has been a wonderful season, with speakers such as Safa El-Khanoussi, Judith Butler, Constance Debré, Anton Jäger, and tonight, Anna Tsing. Each of these speakers has invited us to reflect and engage in conversations. Every conversation was a meaningful encounter between the speaker and the audience. And I would like to express my appreciation to the audience, to you, for making these conversations possible. Tomorrow morning at 9, we will open the ticket sales for the new season 26-27. And with more than 250 concerts and performances, it promises to be another richly packed season, with fascinating and interesting speakers such as Simon van Saarloos, Masha Gessen, and Maria Masha Aliokina from Pussy Riot. You can discover the full program in the seasonal brochure or on our website, thesingel.be. How have fungi shaped human history? At the invitation of the SINGEL and the Flemish Architecture Institute, the Chinese-American anthropologist Anna Tsing will give a lecture on the future of fungi. What has gone wrong in our relationship with the living world, and what can we still change? Anna Tsing is affiliated with the University of California, Santa Cruz, and is an anthropologist and the author of the world-famous book, The Mushroom at the End of the World. Together with Fei Fei Zhu, she curated the exhibition Fungi, Anarchist Designers, at the Nieuwe Instituut in Rotterdam, which is running till August. In this lecture this evening, Anna Tsing returns to the questions that preceded this exhibition. I also want to mention the exhibition We the Bacteria, which will be presented by the Flemish Architecture Institute from the end of September till the end of January. An exhibition curated by Beatrice Colomina and Mark Wigley that argues for architecture that embraces microbes instead of fighting them. But back to the night. Following the lecture, there will be a short conversation between Anna Tsing and Dennis Paul, director of the Flemish Architecture Institute. And there will be also an opportunity for the audience to ask questions. To conclude, I would like to thank the Flemish Architecture Institute for this collaboration, the technical team at the SINGEL, and our production manager, Jessie. Have a nice evening. Thank you.
Thanks. It's a real pleasure to get to talk to you tonight. This image is a wheat rust fungus, and I chose it because it looks like all those alien films where things jumped out of people's chests. And fungi can be pretty spooky that way. So As you just heard, I'm, together with Fei-Fei Zhou, curated a show called Fungi Anarchist Designers in Rotterdam, and it will be open till the beginning of August, so let me encourage you to make the trip. One of the special things about that show is that most of the exhibits were produced through collaborations between artists and scientists. We didn't allow either art for art's sake or science illustration. Instead, we wanted the artists and scientists to be equal partners. And that's one of my messages for this time of catastrophe, that we need to work across many of our differences to get our politics and policies right. Another goal is to show you the importance of working with landscapes, that is, multi-species assemblages in particular places, as units for thinking and imagining futures. It's not enough to appreciate fungi isolated from the worlds in which they live. My book, The Mushroom at the End of the World, happened to come out, or maybe help stimulate, a moment of excitement about fungi. Suddenly, fungi are interesting and maybe even beautiful. But most of the time, this new interest isolates fungi from their landscapes. Either they're lovely and delicious objects, or alternatively, they're ideal building materials, the new plastic. Neither of these foci allows us to think about life on Earth. Let's tilt your interest in fungi just slightly. Let's look at fungi as participants in making and destroying living landscapes. And let's look at fungi within the terrors and challenges of the Anthropocene, this time of human-sponsored environmental destruction. This talk is a prequel to the show at the New Institute. Consider two kinds of fungi. The mushroom at the end of the world tells of the expensive gourmet mushroom, matsutake. Matsutake are mycorrhizal fungi, that is, fungi with symbiotic relations with plants. Mycorrhizal fungi open attention to the importance of interspecies ecologies in the resilience of life on Earth. Through their generous metabolisms, which bring nutrients to beings other than themselves, mycorrhizal fungi nurture forests. They show us the importance of mutualisms in the web of life. But there's other kinds of fungi. Pathogenic fungi, that is, fungi that kill, show us the dangers of the Anthropocene. Working together with human infrastructures, fungi have been responsible for more species extinctions than any other group of organisms. While mycorrhizal fungi support the resilience of sustainable ecosystems involving humans, such as woodlands, pathogenic fungi respond to Anthropocene conditions to undermine sustainable ecosystems, threatening more than human livability. Fungi, more than any other kingdom, reveal the structural roots of the biological catastrophe of the Anthropocene. Many fungi live together with plants, and coordinations between fungi and plants make the diversity and richness of forest life possible. Plant-fungi coordinations can also involve humans, as I once studied in relation to matsutake mushrooms growing in village forests in Japan. Humans raked leaves and cut firewood, allowing matsutake and pine to thrive. Pathogenic fungi are also involved in coordinations with both plants and animals, including humans. And tonight, I'm going to just explore that problem. What kinds of anthropogenic, that is, human coordinations, encourage fungal virulence? How have fungi adapted to the infrastructures of the Anthropocene, becoming some of its most deadly collaborators? These questions reach beyond the world of fungi to give substance to a still developing form of history in which human and non-human pasts are each allowed their full weight. In studying the environmental challenges of the present, we're hurt by the great gap between the sciences and the humanities. That is, the study of non-humans and the study of humans. Most ecological scientists haven't had the time or training to learn about the human histories, social patterns, and events that historians and social scientists show us. Most social scientists and humanists don't have the will or expertise to pay attention to the non-humans. Both kinds of simplification cause trouble for understanding environmental challenges, which are simultaneously human and non-human. We need new habits for the study of both human and non-human histories as they intertwine and make each other. We can't tell human stories without including the stories of non-humans, and increasingly vice versa. In this talk, I'm offering a challenge for you, the listeners. I'm going to tell stories in which fungi rather than humans are the protagonists, that is, the lead players. Since all these fungi move and change together with human projects and histories, humans are not missing. But I'm going to leave out the genres you may have learned to expect from an anthropologist: conversations with quirky people, heroic effects of small communities. These kinds of stories are important, but I'm pushing you towards a different kind of storytelling. I'll tell you of fungi that have become intertwined with human projects. What are their effects on our common world, human and non-human? Is this the future commons we want to build? My protagonists are not crops or commodities, that is, organisms that enact the needs of humans, but they're not wild things. I call them feral, completely dependent on human projects, but also outside of human control. In my terms, feral biologies are biological responses to human infrastructures that, through rapid evolution, adaptation, geographical and interspecies redistributions, allow organisms to set off new historical trajectories. Non-humans are making history together with humans and not in our control. Organisms with feral biologies are not puppets, but they're also not autonomous from us. Feral biologies emerge within the human-made world, but they're notorious for their unruly disruptions. Feral biologies are a key component of the Anthropocene. We need to learn more about them. Let me suggest 4 challenges of Anthropocene feral biologies. First, global spread. Feral organisms often become part of a contagious cross-continental Anthropocene. Feral fungi, my protagonists today, spread through attaching to Anthropocene vehicles in the widest sense of the word, thus becoming much more than localized problems. Vehicles here can mean trains and planes, but it can also mean host organisms, such as humans, which carry pathogens and parasites to new places. It might even mean anthropogenic environments, that is, human-created conditions conducive to that feral biology. Pathogens, like other organisms, have always moved around, but the speed and the scale of intercontinental transfers that have been enabled by imperial expansion and global capitalism are unprecedented. Long-distance dispersion once was slower and more sporadic, allowing forests, for example, to slowly recover from plagues. Constant and rapid long-distance movement along the infrastructures of industrial trade and imperial conquest have created new forms of destructiveness, and these forms are so novel that we identify this movement as part of a striking new epoch, the Anthropocene. In a moment, my first example will offer a strange history with which to think this problem: the spread of dry rot with imperial ships and wood-framed buildings. Dry rot is not a pathogen since it only infects dead material. Compared to many Anthropocene stories, this is a benign one. But it illustrates the principles of the worldwide Anthropocene proliferation of feral ecologies. A second challenge: enabling infrastructures. Anthropocene projects encourage the emergence of dangerous feral biologies. The existence of pathogens is as old as the living world, so there's nothing particularly Anthropocene about the existence of pathogens. And yet, I'll argue, Anthropocene projects, such as the concentration of genetically uniform organisms in simplified ecologies, encourage both the gathering of pests and pathogens as well as the evolution of new forms. In my second example, I'll explore the problem of feral virulence in relation to plantation economies, a veritable breeding ground for fungal pathogens. To give concreteness to this exploration, I'll explore plantation pathogens such as rubber leaf blight, conifer root rot, and southern corn blight. My third focus is the creation of newly virulent pathogens through the work of hybridization and host switching. Such work is easy when closely related species are crowded together, as for example, in the commercial nursery trade for plants, as well as the global trade for live animals. Emergent species and variants flourish in the interplay between the simplified ecologies of commercial agriculture on the one hand and the crowded multiplicity of the industrial trade in living things on the other. While some emergent species and variants are just fine at living with others, this is an opportunity for the development of new forms of pathogenic virulence. The plantation pathogens I discuss in the second part will come up again here, and I add the disheartening stories of white pine blister rust and BD chytrid fungus, the frog killer. The fourth challenge is the emergence of fungicide-resistant fungal pathogens. Consider Candida auris, a sometimes deadly fungus, which has taken up residence in hospitals. Candida auris is practically impossible to eradicate, as it's developed resistance to most fungicides, even in combination with each other. These photos are a bit gross, but I'm trying to scare you because it's serious. Different variants of Candida auris have different drug resistance abilities, and they can exchange these with each other when they come into contact. How might such a situation come into being? By saturating land and water with fungicides, Anthropocene agriculture has made a world for fungicide-resistant pathogens. Worse yet, ordinary soil fungi, many of which are beneficial to plants, including crops, have been killed off. What is this world we are making? Okay, this was my first challenge. How do feral fungi go global? Between the early 18th and mid-19th centuries, the British Navy experienced a serious setback. Its ships began to rot before they could even set out to sea. In his report on this history, John Ramsbottom writes, quote, "The duration of a ship was estimated at 25 to 30 years in the 17th century, about 12 years from 1760 to 1788, about 8 years during the Napoleonic period, dwindling to no duration immediately after Trafalgar," end quote. The problem was the dry rot fungus. It was the death knell of wooden warships, solved only by the introduction of ironclad ships, such as the British Thunderer in 1863. Let me repeat, humans did not invent destructive fungi. Destructive fungi have existed a lot longer than humans, but Anthropocene infrastructures have succeeded in the round-the-world spreading of some fungi that otherwise would have remained in circumscribed places. It's that spread, in many cases, that's caused all the trouble. Dry rot, caused by the fungus Serpula lacrymens, is not a pathogen. It eats dead wood. It's only important to those who happen to be attached to a particular piece of dead wood, for example, if it's part of a ship or a house. It makes a good protagonist for this part of my story only because it shows how fungi have been transported around the world, in this case as part of imperial ships. In its natural habitat, the fungus is a specialist that competes poorly with other fungi. It lives on dead wood in shade at the cool, dry tree line. In the protected environments of human construction, however, it thrives and proliferates. The distribution of Serpula lacrymens has confused geneticists because one variant is so widely distributed across Europe and North America in human habitations. The search was on for its wild relatives. Several scattered relatives were found, but the closer researchers looked, the more different these relatives appeared from the kind that lived in ships and houses. There are indeed wild dry rots, but they're rare parochial and specialized. Furthermore, the kind on ships and houses has a very small genetic range, suggesting recent introduction. Geneticists are wary of political histories, which can't be tested with their methods. However, most authorities have come to accept the idea offered by South Asian fungus hunters in the mid-20th century. Dry rot was transported around the world by the British use of logs from the western Himalayas. As Ramsbottom put it, quote, "Foreign timber was often floated down rivers and then immediately loaded into the confined holds of timber ships, an ideal arrangement for fungal infection. The logs were sometimes covered with fruit bodies before they even reached the dockyards," end quote. From British ships, dry rot toured the world, establishing itself particularly in areas of British settlement, such as North America and Australia. Dry rot thrived with the vernacular architecture of British Empire, which involved wood along with non-organic materials. Dry rot as a method to move across non-wood materials such as plaster. To explore potential food sources, it cleverly makes long cord-like rhizomorphs, which are root-like fibers made from fungal tissue. It excavates useful minerals from plaster. It travels from one part of the house to another part, and from house to house. Wood frame cellars are perfectly designed for serpula, and whenever house styles came to use them and the temperatures were cool, dry rot came along. Dry rot was a companion of British colonial culture. In learning to occupy a world where the sun never set, it changed from an aid to local forest succession, making nutrients available for a new generation of plants, to a superhero of destruction, leaping buildings in a single bound. Dry rot responded to Anthropocene proto-industrialization, the global deployment of standardized products from ships to timber. Its history shows how feral biologies emerged with new forms of travel. Ships took dry rot from continent to continent. Settler houses provide delightful new habitats for the fungus. But transportation is not the only form of Anthropocene attunement through which feral biologies thrive. To explore new capacities for growth and reproduction, it's useful to turn to the production of resources in simplified, disciplined ecologies, that is, plantations. We know plantations best for the engineered crops we made for them, the miracle grains, the fast-growing trees, the genetically modified soy. But plantations also became breeding grounds for feral biologies, and these show us more about Anthropocene dynamics than all that showy but vulnerable engineering. Engineered crops need lots of work to maintain. Feral biologies, in contrast, take care of themselves. Plantations are feral incubators. Anosum root rot, a white rot resulting from a group of cryptic species of the genus Heterobasidion, is the most feared resident of modern conifer plantations. From the perspective of commercial owners, it causes billions of dollars of damage every year. Unlike serpula, Heterobasidion can feed from live wood as well as dead, and it's virulent enough to take down whole plantations. But Heterobasidion does different things on live and dead wood, and its ability to switch back and forth between necrotroph, eater of things it kills, and saprotroph, eater of things already dead, is key to its ability on the plantation to transform from humble fungus to serial killer. Here's the story. Heterobasidion spreads in two ways. In primary infection, a spore germinates on a freshly exposed stump. It wants dead wood. In secondary infection, mycelial growth moves from the live roots of one tree to closely related others through the natural grafts formed by roots under the soil. Outside of plantations, neither infection route causes much forest damage. Heterobasidion has trouble moving very far. Instead, it stimulates succession by killing off a few trees, allowing others to replace them. In a plantation, however, Heterobasidion has an enhanced ride. There are stumps from felling side by side with closely related trees, perfect for a mass of root grafts. The plantation thus makes a new world for the fungus, a world where nothing stops it. The variety and patchiness of woodlands, which kept Heterobasidion humble and small, is replaced by one great womb for Heterobasidion spread and reproduction. From stump to stump and from root graft to root graft, the conifer plantation seems designed for the unlimited reproduction of the fungus. Indeed, Heterobasidion is not alone. Think of any plantation crop, and there's a fungus that has been remade as its champion killer. A devastating incident of southern corn leaf blight makes this point even more directly. Fungal virulence is stimulated by the political ecology of the plantation. The narrow genetic stock that makes plantations easy to work with for capitalist economies also makes them breeding grounds for fungi, which develop variants to infect just that genetic stock. Consider corn, where breeding practices created plants so genetically similar that their height and time of ripening is standardized, allowing mechanical harvesting. The genetic similarity of these crops has meant that fungi evolve the ability to infect, that if they evolve the ability to infect one plant, they can infect them all. The fungus that causes southern corn leaf blight was a minor pest until 1970, when it managed to kill 15% of the corn crop of the United States, as well as corn across all those parts of the world supplied by U.S. hybrid seeds. Journalist Jack Doyle described the episode. The new fungus moved like wildfire through one cornfield after another. It could wipe out an entire stand in 10 days. Whole ears of corn would fall to the ground and crumbled at the touch. The fungus moved swiftly through Georgia, Alabama, and Kentucky, and by June, its airborne spores were headed straight for the nation's corn belt, where 85% of the all-American corn is grown. By this time, however, unsuspecting Corn Belt farmers had already planted their crops and were largely unaware of the bitter harvest headed their way. The astonishing rise of southern corn leaf blight turns out to have been enabled by a genetic modification that caused male sterility in corn plants. Corn's pollinated by the wind, and this means that farmers cannot control the parentage of their corn ears as long as pollen from varied corn plants is around. Between the development of high-yielding hybrid races of corn in the 1930s and 1969, farmers who planted hybrid seed hired workers to remove the male flowers of corn, called tassels, by hand. In the 1950s, however, U.S. agronomists had found an abnormal plant that was already sterile for male flowers, and they developed it through back-crossing into their hybrid seeds. Since industrial planting practices homogenized seed sources, most all the corn in the U.S. Corn Belt and much corn in other countries use this single genetic modification. This genetic homogeneity laid out a feast for the right fungus. When the fungus developed a variant able to infect plants with this modification, every plant was stricken. These were the conditions for making a plague. Agronomists quickly understood the problem. Unfortunately, seeds for the next year's crop had already been sold both in the United States and around the world. Thus, another year of blight ensued. However, subsequently, the particular genetic code for male sterile corn seed was withdrawn, and this fungal variant was no longer a problem. Yet this quick action was only a success in Anthropocene business as usual. While many scientists spoke of the need for maintaining genetic diversity in crop fields, this did not happen. Another strain of genetically homogeneous corn became dominant, setting the stage for another fungal blight in the future, as soon as the fungus finds the way to infect this one. This has been the practice, too, for the many fungal rusts and blights infecting other commercial crops, from wheat to bananas. Offer lip service to the importance of genetic diversity, find a resistant variety, and then continue monocropping practices as before. Critical discussion of the Anthropocene. should instead block such business-as-usual frameworks. To take seriously the planet-wide dangers of the Anthropocene might mean to question the plantation. Next challenge. What does it take to become an Anthropocene killer? Plantations are useful to think about the scale of fungal plagues. Some fungal pathogens are limited to particular plantation crops, while others spread widely and jump from host to host. This difference matters. While the former are merely an annoyance to plantation owners, the latter destroy living arrangements for many ecological communities. The difference often depends on commercial travel trajectories, as these are involved in the spread of plantations with their pathogens. An example of a pathogen that has not yet traveled is rubber leaf blight, which continues to be found only in the Amazonian homeland of the rubber tree. When Henry Ford opened his failed rubber plantations in Brazil, which he called Fordlandia, in the 1930s, he stimulated the rapid reproduction of rubber leaf blight, which destroyed his rubber production dreams. The fungus did not have to develop a new variant. Merely planting genetically homogeneous rubber trees so close that their leaves touched allowed asexual spores to spread from one tree to another, killing off the plantation. The presence of the fungus in Brazil accounts for the fact that rubber plantations continue to be found only in Africa and Asia, where the fungus has not yet traveled. In contrast, the root rot I mentioned, Heterobasidion, has begun the process of travel through the movements of colonialism and industry. As conifer plantations are carried to new parts of the world, so is Heterobasidion. Under conditions of global resource mixing, plantation conditions create new powers for Heterobasidion. They allow variants to get together, forming new fungi with new powers. Heterobasidion has an extremely long genome with many transposable elements, and the new hybrids affected by resource mixing seem to have activated such elements in ways that allow them to pick on new hosts. This slide shows the mechanism through which hybridization enables host shifting. In Western North America, the bringing together of Heterobasidion irregulare and Heterobasidion occidentale in pine plantations has created the ability to attack sequoia, which was previously unaffected. In Italy, the appearance of North American Heterobasidion irregulare along native European Heterobasidium annosum has managed to bring the pathogen into oaks, not a conifer at all. In this form, it's begun to spread across the natural forests of Italy. Heterobasidion has begun to map out a truly global terrain by forming combinations that attack new hosts and reshape landscapes both inside and outside the plantation. New abilities to travel with and without human assistance. This might be the key characteristic of more than human Anthropocene proliferation. The architecture of plantations creates biologies of virulence. As plantation practices spread, they create feral ecologies that spread without human assistance. When plantations are combined with global resource transfers, the feral organisms that rise up from these arrangements may gain superpowers that make it almost impossible to imagine blocks and controls. When landscape managers confuse the plantation and the world, plantation-borne plagues begin to spread indiscriminately. The mixing of different variants of Heterobasidion has allowed the fungus to spread across non-plantation landscapes, killing new kinds of trees. This is a plague and hard to control. Yet Anthropocene business-as-usual strategies have been brought to this task, fighting plantation plagues by spreading the plantation. It's worth spending some time with what happens when the world is treated as if it were an agribusiness plantation. One example involves the spread of white pine blister rust in the United States. White pines are indigenous to North America, and in many places they grow quickly on abandoned fields. Eastern white pine started the U.S. timber industry. And when those white pines ran out, the industry cut the Midwest. And then when those pines ran out, other pines in the Northwest. At that point, late 19th century, white pine plantations on cut-over land emerged as an economic crop. From this point on, plantation pines and naturally occurring white pines intermingled in pine country across the United States. The confusion between plantation and cut-over land and forest, the confusion between plantation and forest became a mainstay of the U.S. timber industry. Forests were managed as plantations, and cut-over lands became both plantations and forests. Blister rust was introduced, managed, and spread through this confusion. Plantations wanted seedlings, but American nurseries were getting much better prices for ornamental plants. Planters looked to Europe, where American white pine had been introduced. Tariffs were removed, and by 1909, millions of white pine seedlings were pouring into the U.S. from low-cost nurseries in Central Europe. White pine is not indigenous to Europe, and the nurseries mingled introduced pines from many places. Thus, these cosmopolitan nurseries contaminated their seedlings with an Asian fungus, white pine blister rust. As soon as they saw it killing their pines, Americans were alarmed, and by 1912, Congress passed its first quarantine law to stop the trade. But they were closing the barn door after the horse had left. Confusing plantations and forests spreads feral fungi. Planting infected seedlings in or near natural forests spreads the fungus throughout the forest. Both planting and disease protection require lots of labor. In the case of white pine blister rust, the U.S. Forest Service made an all-out effort to control the disease. However, in the end, it wasn't enough. There just wasn't enough labor to treat forests as well-managed plantations, so they instead were treated as poorly managed plantations. And while rust-resistant varieties of eastern white pines have been developed, for many western pines, they have suffered. White bark pine has almost been wiped out by this traveling fungus. Loss of life forms cross borders these days. Lots of life forms cross borders these days. It's easy to imagine that such transfers are just a natural part of human travel and trade. Perhaps we might think they're a necessary concomitant to freedom. After all, are we all supposed to stay at home? This response, however, neglects the conditions under which feral biologies develop and spread. This is not just travel, it is industrial travel. If this is freedom, it's the freedom of capitalists to change and destroy life. Capitalist freedom also leads us to the mass death of frogs and other amphibians. The frog-killing fungus, popularly called BD, as well as its salamander-killing sister, have spread with commercial amphibian trade since the 1970s. A particular variant, which scientists call the global panzootic lineage, has proved really virulent and widespread. It's spread since the late 20th century with the expansion of the global amphibian trade for exotic pet, medical, and food purposes. Commercial shipping crowds frogs together in unhealthy conditions. Diseased frogs are released in new places, and the frogs carry the fungus to new populations of frogs in their destinations. One of the most common carriers of the disease is the American bullfrog, which has been heavily promoted by international development experts for rural enterprise. This promotion of American bullfrog farming in many areas of the global south has allowed bullfrogs to escape from their farms to infect indigenous frogs across the countryside. For example, American bullfrogs were imported en masse to Brazil for small-scale development projects in the 1970s. Dramatic declines of wild frogs in Brazil began in 1979. These declines were not on frog farms, but rather far beyond them. American bullfrogs had long left the farms, establishing themselves throughout the countryside. They travel everywhere, they eat everything, they reproduce avidly, and they're bigger than most other frogs and easily expand into their spaces. it seems likely that American bullfrogs not only ate other frogs, but also infected the survivors with BD. The most frightening thing about BD is that it kills frogs in the most remote areas, far from the plantation-like frog farm. BD makes use of the flow of water and the sociality of frogs to find its way to places where we can hardly think to anticipate less control. Furthermore, frogs travel the world not only as commercial cargo, but also as hitchhikers in other industrial supply chains. Asian toads recently traveled to Madagascar, hidden in mining equipment. Other aspects of industrial supply chains make frogs more vulnerable. Fossil fuel-driven climate change makes the world hospitable for BD. Low-level chronic pesticides have made frogs easier to kill. At the intersection of multiple capitalist forces, BD has become a global killer. The resulting plague is a major extinction event of our times. Our grandchildren will inherit a world deprived of frogs. Saturating the world with fungicides makes way for more powerful killers. The simplified ecologies of commercial agriculture and livestock rearing gather fungal pathogens and create opportunities for rapid evolution of new ones. Saturating the environment with fungicides exacerbates this situation, encouraging the development of resistance and thus new forms of virulence. Fungi are flexible and opportunistic. Many of the deadliest are capable of rapid evolution, whether through mutations, hybridization, gene exchange, or relations with new hosts. They take advantage of new resources and new niches to remake themselves to survive and thrive. Meanwhile, the range of poisons we can create to knock back fungi is limited because their metabolisms are similar to ours. Most current fungicides take advantage of the cell wall chemistry differences between fungi and animals. But this offers a limited set of options compared to, say, bacteria. Fungicide resistance cannot easily be overcome by the development of new fungicides. Perhaps you've heard that banana plantations are in trouble, attacked by the unstoppable fungus, Fusarium oxysporum. Fusarium have mechanisms to survive fungicides. The variety called TR4 creates asexual spores that persist in the soil, which germinate to cause further infection. This ability is encouraged by fungicides. Plantation bananas all over the world have died in droves, researchers explain. There are currently no commercial banana cultivars resistant to TR4. Many Fusarium have an intrinsic resistance to fungicide, but they've also picked up new forms of resistance after exposure to fungicides. We can't depend too heavily on differences across species and subspecies in this genus because, most excitingly to a biologist and most dangerously to any organism about to be infected, they've shown themselves capable of transferring genetic material through horizontal gene exchange, that is, without bothering with sex. Entire chromosomes have been transferred across varied strains of Fusarium oxysporum, indeed enough genetic material to account for 1/4 of the genome. Non-pathogenic strains become pathogens through such transfer. It seems likely that both intrinsic and secondary resistance can also be transferred. And don't be too dismissive about the plight of the bananas. Some strains of Fusarium oxysporum infect humans. Indeed, approximately 10 species complexes of Fusarium can infect humans. Fusarium species are heralded in geniuses in trans-kingdom pathogenicity. So humans are in the animal kingdom, bananas are in the plant kingdom. So trans-kingdom pathogenicity means the ability to sick in living beings across this wide gulf of kingdoms. Pathogenic strains switch hosts, developing further agilities. Resistance to fungicides is only one of the many abilities of this fungus. Aspergillus are a group of molds that live in the soil where they eat dead plant material. They're not ordinarily pathogens. They mostly reproduce through clones, but they're capable of sexual reproduction, and at least some have characteristics they call panmixia. In other words, they can mate across all boundaries, quickly spreading new traits such as virulence and fungicide resistance across regions and environmental niches. Aspergillus came to medical attention when an increasing number of infections to humans were reported, and increasingly, too, were found untreatable through medical fungicides. Like Fusarium, they've learned to jump kingdoms to infect living beings as pathogens. This is the group of pathogenic fungi for which connections to the rise of the use of fungicides in commercial agriculture are most clear. Studying Aspergillus fumigatus involved in human infection, medical researchers found two genetically different kinds of resistance, one among patients who'd been prescribed long-term doses of fungicides for whom resistance could be attributed to the fungus changing to fight that treatment, and another among patients with no previous experience of antifungal treatment for whom the infection must have come from the environment. The meteoric rise of fungicide use in agriculture corresponds closely in place and time to the rise of fungicide-resistant Aspergillus fumigatus infections. The same resistant strains found in patients were found in flower beds. The infective Aspergillus were resistant to azoles, the very fungicides most used not only in commercial nurseries and fields, but also in paint and coatings, wallpaper paste, clothing, and wood preservation. Azoles pervade not just agricultural soil, but wastewater and sewage sludge. Aspergillus, saturated in azole fungicides, developed pan-azole resistance by 2007. A soil fungus became a lung fungus and almost untreatable. The infective yeast Candida auris hit the medical world by storm because no one knew where it came from. Candida auris, first identified in 2009, is considered an emergent pathogen, and it's resistant to most fungicides. One of the strangest things about Candida auris is its almost simultaneous emergence through different routes in 4 different places and with 4 different genetic signatures. What happened around the world to allow this dangerous fungus to emerge? Candida auris distinguishes itself by its ubiquity in hospital settings, where no one can get rid of it. One research group explains, quote, Candida auris contamination has not only been seen on bed rails, bedpans, mattresses, linen, pillows, furniture, door handles, flooring, walls, radiators, and windowsills. It can even spread as far as bathing areas, sinks, mop buckets, and cleaning equipment. Medical equipment that comes in contact with the patient also gets readily contaminated. For example, temperature probes, blood pressure cuffs, glucometers, housekeeping cups, alcohol gel dispensers, dialysis equipment, ultrasound machines, computer monitors, keypads, and cell phones. That's the end of the quote. This ubiquity suggests that saturation with antimicrobials in the hospital as both medicines and cleaning products may also be a source of Candida auris' resistance. This hospital story forms just one part of what researchers Chakrabarty and Sood call, quote, the crucial ecotoxicological disruption, the overwhelming saturation of our biosphere with biotics and antifungals, end quote. This is the route taken by Alex Liebman and Robert Wallace in my collaborative project, Feral Atlas. They argue that Candida auris is a factory fungus, that is, a concomitant of the ecological simplifications and toxic chemicals of commercial agriculture, like the pathogenic forms of Aspergillus and Fusarium. Might we be actively building a world for fungal pathogens with resistance to fungicides? The more than human Anthropocene. Pathogenic fungi have become collaborators in building the Anthropocene in all its dangers. Let me repeat my 4 challenges. First, feral fungi travel the world with Anthropocene vehicles. Second, they're assembled and encouraged in plantations and other Anthropocene infrastructures. Third, they evolve and adapt to become more attuned to human environments, and in the process, they become more dangerous. Fourth, Saturation of the environment with fungicides encourages the rapid evolution of fungicide-resistant strains, which threaten the health of humans as well as other Earth beings. We have encouraged this. We are in trouble. Attention to this partnership in destruction is the beginning of another kind of collaboration, one that's necessary to what I've called collaborative survival. Instead of fighting for survival as individuals, we need to develop allies, human and non-human. This can't be done without knowledge from both the sciences and the humanities, and it also requires political policy, attention to the death-dealing collaboration of pathogenic fungi in industrial farming and shipping is a place to begin in forming this alternative collaborative network. We should also remember other alternatives, such as the mutualist relationship between farmers, mycorrhizal fungi, and trees in Japanese village forests, which might form a model for more than human alliances. Again, this is a part of a diagram I made with artist Elaine Gann to illustrate the mutualisms between fungi, trees, and farmers. If you're disturbed by the lack of human protagonists in my lecture, let that be a challenge. I've exaggerated to make the point. Can we tell stories in which non-humans play a central role in making our common history? These are not stories without humans. But I've let human projects be the backdrop to show you the constantly shifting work of feral fungi, and especially some of the most dangerous ones. We should not sit back. Too often we take Anthropocene infrastructures, from plantation monocropping to global shipping, for granted. It's time to refuse this naturalization. We need to act against these forms before they kill us all. Thank you.
Yeah, thank you very much, Anna, for this beautiful travel through fungi, spores, roots, leaves, plantations, sort of histories through our Anthropocene, taught through feral biologies, basically. I would like to circle back to the beginning of your lecture, where you said this is a sort of preludium to the current exhibition at the new institute. And to what extent working with artists, you mentioned before the collaborative dimension that also maybe forms a kind of a basis of resistance for new alliances. So how does the working with artists maybe allows you to shape those new alliances, or to give visibility to those new alliances as well through this work?
I myself was really impressed at how each of the teams figured out a form in which to tell their story that I don't think either the artist side or the scientist side would have done on their own. In fact, in some cases, I saw, rough thinking from the scientist side, and it's completely different than what came out at the end through this collaboration. And so it was really inspiring for me to see the stages in which collaboration itself created something. For example, a collaboration between a biologist named Rob Dunn and an art team, Bauman Leahy, created an exhibit about the yeasts that help us digest our food inside the human intestines. And they've been interested in yeast, but the original idea of the artist was, the ways that humans over time have made beer and, all the different kinds of ways that fermentation with yeast is important. But the biologist had done all this work on how yeast themselves work and human evolution in relation to yeast. And so in the end, you get this exhibit that's the human intestine from the perspective of yeast, which I think is a very fine way that neither the artist or the scientist might have come up by themselves. So it's just one example.
Fantastic. In a way, it allows new forms of imaginaries, basically, that other domains or singular domains are not able to imagine by their own practices. As a historian, I'm also curious about the time spans we are talking about when we're talking about that fungi develop from let's say, from one state being attached to a host, becoming maybe a more a fungicide species, which also kills. And in a way, you mentioned this, the dead rot example, basically that the dry rot example that traveled through ships, distributed to the colonial routes, and became, let's say, a massive fungi when it came to timber construction all over the place. And I guess while introducing other building materials, plaster, concrete, bricks, I guess this also led to a symbiotic type of transformation of the fungi itself. What kind of, how long do we need to imagine this type of transformation?
Yeah, I think none of us are surprised when evolution takes a long time, and certainly it can. And especially a serious symbiosis, that means two organisms that are able to figure out a way to live together through their relationship to each other. So that takes a very long time. But what's really shocking to me as I've tried to read more about these dangerous fungi is how short the time period is. I mean, I think Candida auris, I think, was just discovered for the first time in 2009. And it had developed, as I was saying, 4 different races already that are quite different than each other, but somehow converged in this moment of the overuse of fungicides in agriculture. So what's really shocking is the kind of quick turnaround through which these fungi are able to make use of their own, evolutionary abilities, but to come up with new combinations that latch right on to these industrial infrastructures.
Yeah, I guess it's pretty convenient or evident when you talk about how pesticides work in plantation fields. But what was for me really stunning is the example where you mentioned about this Candida auris, which was in this hospital environment, that basically points out also to the fact that sort of our more and more hygienic and sterile way of living, and hospitals are definitely an example where the microbial resistance itself has raised enormously, because only certain type of microbes are only able to survive within there because of our kind of overuse of,
so
of hygienic products.
Only the deadliest ones, in some sense. Or, I mean, and that's also true in a field when we spray it with deadly poisons. Only the very worst microorganisms stay alive, the ones we least want to work with, and all those beneficial ones are being killed off.
Well, I guess those should be an alarm to us about our extractive use of the environment and our way of basically an almost unbound way of capitalism that sort of kind of eradicates everything around us, and only those microbes should be the alarm signals that, or the fungi should be the alarm signals that basically point to our excessive use of those.
Yes, I agree, and that the whole kind of philosophy of singling out one kind of organism to be your crop, and very closely related examples of that organism, and to stuff them together so closely, it's just asking for all kinds of trouble.
All right, staying with the trouble is maybe also one thing.
Well, not in that sense.
Not in that sense,
yes,
I guess.
Asking for all kinds of danger for life as we know it.
I'm looking in the audience whether there are any questions. I see one question over here, another over there.
Hi. Well, thank you very much for the talk, Anna. And it was really a great introduction for me to realize the impact of... So 2 words kind of stand out for me, like homogeneous and scale. And I was just curious to know, like, what's your relation... Like, if we create ecosystems that become heterogeneous, so I'm thinking about alternatives way of agriculture, like permaculture, like I'm curious to know is that a solution for you, like to create this ecosystem, and what's your relation? Do you have a garden? Like, I'm just curious about these alternatives ways of resisting towards industrializations of food and this.
Yes, so everything you're saying, let me just push it a little further, is that humans have lived on Earth for a very long time without causing necessarily all these problems. And we see, the case of coffee in Central America, as studied by Yvette Perfecto, is just so striking to me, because peasant coffee was a polyculture coffee. That coffee likes to be in the shade, and you have tons of other things going on. It was a very healthy coffee. And then the pressures of the industry made them just do this full sun monocrop thing, which then ended up killing off all the coffee because of a fungus. And it's a kind of parable for our times, in a way, that there are plenty of other ways of doing agriculture as humans that don't cause the same destructive effects.
Yeah. Okay. So the monocrop aspect is really the kind of the core of the issue.
Destroying the soil, so that because of no respect for the kind of life of the soil. So between deep plowing and all of those chemicals that they're putting in it, the soil itself stops being able to support plants.
Okay. And do you know Fukuoka?
Pardon?
Do you know the person who wrote The One Straw Revolution?
I know of the book, but I don't know the person personally.
Okay.
Yes.
Thank you very much.
Hi, Anna. Thank you so much for your talk. I am Danish, and I'm a PhD researcher. One thought that came to my mind was that you were talking about the mutual relationship and the interaction between human and non-human. How do you position your approach to be different from the One Health approach, which is becoming a buzzword after the pandemic? And do you feel that the challenges that the One Health approach is bringing to us also comes with your conceptualization? The second question that I was thinking of from the challenges that you talked about, and I'm a lawyer, so I was looking at the plant quarantine laws that you talked about. Would you consider adding lack of governance, in the sense, because the governance is fractured and it's very human-centric and doesn't think, I'm not saying think like how the fungi will think, because we don't know, but so how do you think about that this is another governance challenge? And I feel that the way that you talked about history was that there has been reactions to what has really happened, and there is no preemption to what might come in the future. Even though we know that capitalist structures have made and rebuilt and reacted to what the fungi has given to us. So I was wondering of this challenge of governance and on the One Health question. Thank you.
I'm not going to give a good answer on the One Health thing, because I haven't been thinking about it recently. There are some things we really need One Health for thinking particularly about all these zoonotic diseases, that these same kinds of ecological simplifications are just making it so, you have put thousands of chickens in close display, and you're going to develop all these new avian flus. So in that sense, we really need it. I think critical scholars have argued that sometimes One Health doesn't do enough to think about the social context of health, the political economies within which health is organized, and that if we want to understand virulence, for example, like why some microorganisms become so deadly, it's not going to just be about the nature of the organisms. It has everything to do, as I've been trying to argue here, with the kinds of social and economic contexts in which these biologies develop. And so I know there's been some attempts to push One Health with some difficulty in that kind of direction. So that's at least one tiny piece of it. governance is a marvelous challenge to try and think about in terms of this attention to how humans and non-humans live together. And there's so many different things that one could say, from all of the very interesting experiments that are going on in some different parts of the world. I mean, even, giving rivers kind of personhood and attempting to figure out ways. I mean, I think some of it has to do with legislation that allows indigenous groups to maintain a sense of kinship across species lines, as it were, that these are some kinds of experiments in some places that have some interesting things. But, where I'm doing my research right now, part of the problem is that law is used to further corruption and make the destruction of both indigenous people and species easier. So one has to worry not just about what law says as a set of texts, as it were, but also as a set of practices in which a certain kind of state authority is given to a forms of expertise. So it's a tricky field. It does not mean I want to give up on it, but I need lots of help, maybe from people like you, in thinking about how to deal with not only the fun exercise of thinking about sort of conceptually, how could we bring in more, but also practically, how do we deal with these legal systems that work against both humans and non-humans?
Thank you, Anna. First of all, thank you so much for this evening. I want to respond to the final point you were raising about this historically specific urgency of telling stories in which human subjects are not the only decisive ones. And I want to bring attention to the fact that, or like my experience, that the narrator of those stories in practice are not outside the diagram, the relations, the relationships with the capitalist infrastructure in their very task of narration of the stories. I have to think of what Fred Moten wrote about logistics, right? That the first mass transportation of things were, like genealogically speaking, transatlantic slave trade, and maybe the descendants of those racialized bodies, transported through what we call logistics now, are the artist and anthropologist producing art and knowledge. And I sometimes cannot get rid of this impression that the assemblage of cultural production is a bit like plantations, and I don't think academia is also outside that assemblage, definitely. So within this slide, I want to raise a question. I mean, also, I think I've been personally exploring this, and how do you, like, as an anthropologist who is still dependent on the very milieu that you study, remain feral in your work?
So you're raising really good points, and I would never want to claim that I or other kindred spirits were outside of the structures of power, that we are responsible. I mean, it's not just that we, our daily bread, as it were, comes from those structures, but also that we hold, I'm a U.S. citizen, and it's just a very embarrassing thing, a shameful thing to have to sit in front of the stage and say that at this particular moment in history. And, but I do not want to disavow that whole structure of power. I think So for me, there's something about imperfect as it is, the practice of getting to know other beings and other human beings that allows one to at least begin to seep through the kinds of assumptions that are drilled into us in an everyday way. And that so I do think that some kind of critical awareness and attempt to get closer to what else is going on in the world is possible. Lots of my colleagues and students, for example, think that you should only study people who you're genetically related to. But, I'm a mixed race person, and I wouldn't even know how to get started on that. And so, Anna, I think it's really important for all of us to tell stories both in and out of the experience of our ancestors, that we're doing our... So I don't have a clean answer to you, but this is maybe back to holding on to the possibilities of learning more than what you were brought up with, and that what you're asked, not just brought up with, asked every day to repeat. And I guess I had wanted to say something when I was thinking about how I presented you guys a talk full of kind of deadly stuff, that I think that socially organized anger is encouraging, empowering, helpful, hopeful, that it's part of how we build new futures, is learning from all the ways that we have to trespass what we're asked to do, to be angry, to hold on to our, because we can't get out of them, some of this kind of social status that we're in, and be incredibly angry about it in a productive way, working with other people. So maybe that's at least a beginning.
Thank you, Mrs. Tsing, for a wonderful lecture. Can I continue what you've just said and ask how you've experienced your work as an anthropologist, which is, if I can say so, primarily descriptive, is finding patterns in what you see in history, and your recent work as a curator. What have you learned from leaping into a field which is about creating new things, creating new thoughts and imaginaries? Because indeed you presented us with quite a lot of disaster to come, but making exhibitions with artists and scientists is offering, I assume, I haven't seen the show, but I'm planning to go. Can you say something about that relationship and how those two relate in your work?
I'll try. I think the word ethnography, which I continue to endorse, is both a research practice of trying to really take a slow route to figuring out what other people are up to and how they might teach you to be a different person. But the term ethnography also means writing or expressing or representing something. It's a communication skill that's about trying to then share what it is that you've been working on in some way that might move people. I remember in graduate school hearing the statistics that most journal articles in professional scholarly journals are read by a maximum of 5 people. And I thought, oh, here I am training in this field that no one other than 5 people will read me. So I've tried, not just by myself, but with many others, to create a different kind of practice of expressing what we know. Because I continue to think description is really important, because we know there's so many things we don't know anything about, and that we get told the same stories over and over again without getting to hear about these other things that are going on that we should know about. And so I feel extremely open to all the ways of storytelling or making through which, and it was a thrilling experience to get to do the making part of it as well as the storytelling part in this exhibit, where I got to see these exhibits come to life. I mean, over quite a long time, where they were discussed and thrown out and redone. And so, a long period, and then, big ideas thrown away because we didn't have the money, or we didn't, it wasn't working out, and then reformulated, and all of that stuff that goes into making, down to the final thing of like, should we rehang this banner because it's kind of crooked? That all of those pieces of it were so amazing to me to produce something that had a kind of materiality in the world that's different than what I had gotten to do before. So I don't know if that's the answer to your question, but it was an amazing experience for me.
Thank you. It's a brilliant answer. And one of the key things in addressing the present day is finding new terms and vocabularies and images. Did you find new terms?
From the exhibit?
Yeah, so because I'm teaching and some of my students are in the audience here, actually they gave me your book to read. That's why I'm here. So talking about nice ways of reciprocity, this is how I happen to be here. And I'm very grateful for that. Because one thing that is key in teaching is finding shared terms for things we do not understand yet. My students are learning architecture, and we're facing a world that is quite complicated, and many of them actually doubt a future in architecture. And they are expressing a kind of desire or an urgency to find new terms and new vocabularies because they cannot picture the world they're moving towards. And so making a show is providing us with pictures that we might not have seen yet. Mm-hmm. And so we need to find a language to talk about them. So I'm really curious what you found out in terms of like maybe new notions or verbs or I don't know.
I'm not sure I can give you the big picture, but maybe it's fair to start with the little ones. I mean, from things that particular contributors offered, that architect Phil Ayers was one of the contributors, and his exhibit is called Architecture Must Rot. And so it's about, incorporating fungi into architecture rather than trying to keep them out. And indeed, all this, then people kept showing me things that they're doing in Japan, for example, to use mycorrhizal fungi to inhibit the kind of wood-rotting fungi in houses. And so people had a lot of cool ideas about that, but other tiny things that you wouldn't think would be very important. It turns out that in Dutch, the word for a back way to get from one part of the exhibit to the other part of the exhibit, it's called an elephant path. And it just became a big theme for us to build an elephant path. And we have this whole display of elephant poop with mushrooms coming out that's a kind of attempt to think about alternative paths and back paths and ways of thinking in which, the fungi are leading the way. So I'm sure you're going to have to give me more of a chance to think about what are the big pictures, but I am bubbling up with small ones like that.
Hello, hi. I have a question, whether in your research or through your research on the concepts of ferality, whether you were considering what does that mean for the human monoculture, which is human settlement, the city, because we are here all together in one monoculture group together. So I wonder, how does that respond to, well, how the ferality functions in the situation?
Yeah, my team swore. I have a lot to say about humans, but I'm not going to use the term feral. That was what our agreement was, and I think it makes sense because there are lots of ways that humans have been denigrated, usually on lines of, race, class, and gender, and we don't need to add another dirty word to that. And perhaps there's a literature on feral cities in which it's used in an incredibly negative way. That is not what we mean by feral, that the wildness of the city, gangs, or, dirtiness and things like that is called its ferality, and that is not our usage of the term. So we wanted to get as far as we could with that, but also the very positive use of the term to mean kind of unbridled masculinity. And so both the negative one and the positive one we're trying to stay away from by not using that particular term about humans. But it doesn't stop you from working with these feral biologies to think, what are the implications of, piling up humans in particular kinds of ways and trying to create these, single species enclaves where other species are not particularly welcome? What kinds of worlds are we creating with that? So I think you can ask those questions, but I would rather not use the term feral for them.
All right. Thank you very much, Anna,
Okay.
for this really inspiring lecture
Okay.
and conversation. I hope you enjoyed it as much as I did. As a last hint, please visit the exhibition at the New Institute.
Yes.
It's on until
August 8th, I
August
think, or something
8th,
like
so
that.
you still have a few
but
weeks.
Please do take a look if you have a chance. It's not that far away.
Thank you very much, and I wish everyone a
And thank
nice
you.
evening.
I want to say, because there's these lights that shine in my eyes, when I was actually doing the talk, I couldn't see you guys very well, but it doesn't mean I wasn't grateful that you were sitting out there and trying to listen. So thank you for hanging in through the talk.
Thank you very much.
Thank you.
You've been listening to the SINGEL Recordings, a podcast by the SINGEL. This episode was made by Jessie Bervoets and Maarten Buil and carefully recorded by Christophe van Kranenbroek. For more lectures, interviews and debates, visit our website and discover the full program.